Okay, good morning, everybody. Hope you're enjoying the weather. Thanks. I know my trees weren't enjoying it. I woke up to see a handful of branches that couldn't take it. Not good. I'm like, geez, I'm glad we still had power. Okay, so a couple of quick things. I have not had a chance, well, technically Proctor Free has not completed all of the evaluations. It takes, even though it's computer, it takes time when it's got 50 some odd people trying to evaluate all of that. So I expect that it should be done probably by later today. Scores were not quite as good as I was expecting them to be. The average was lower than the first test. It was 66 or 67%. Not so good. There, as far as curving, I can't tell you exactly where that's going to be at. The last one, we took it as a 93 as the high score. It's going to be more points than that. But not going to tell you any of that until Thursday. After I've had a chance to evaluate stuff. Just looking at numbers real quick, I still need a little more details. Do you guys have questions or concerns for me? Silence. Okay. Anybody tuning in from home? Any questions or issues? Yeah, question back there.
Yeah, how do you calculate your grade for sure? Like, out of like both classes, because
Out of the lecture and the lab together?
yeah.
Okay, so that's a good question. So to calculate your grade, and I will have this stuff. It's not going to be posted on Moodle, but I will be submitting grades. It will be for everybody, whether it's poor or not. I think college, I don't know if the whole university has done it, but definitely College of Science and Engineering wants everybody to know whether it's an A or an F where you stand. So back in the day, it was just poor scores. All right. So you will see that it should post on Bengal Web, I believe. And if your score is poor, then you get some sort of notification. As far as calculating your stuff, you should, and I have not seen lab scores yet. I was told that they would be to me on Thursday, would be when I have all of that stuff. So you should, I believe, be able to see your percentage in lab right now. Is that true?
So it doesn't show like the final percentage. It just shows like all the course or like all like the course and pre-quizzes. It
Okay.
shows the grade for that, but it doesn't show like an overall.
Overall?
Yeah.
Okay, so that's a little bit of an issue.
Yeah.
So I don't know, because I haven't looked at the lab syllabus. I don't know what your overall points are for the lab. My gut tells me it's probably near 500, but I'm not sure. You should know that. It should tell you on there, there's X amount of points available, all right? So it is kind of dumb that it's not showing all of that stuff, but if you know you have done 6 or 7 labs, however, I don't know for sure how many we are at right now, 7, right? So if each of those labs, pre-quiz and post-quiz, because that's all you're really getting points for, right? Pre-quiz, post-quiz, how many points do you get for each of those? Anybody? 10 for pre, 20 for post, so 30 points per lab, right? So you've done 7 labs, that should be 210 points total that you have if all 7 labs are on there, which I'm assuming that's what I will be getting. So if you can add all of that stuff up, whatever you get, divide it by 210, that's your percentage in the lab. Whatever that number is, and then you can see the lecture stuff, you got 2 scores on that. Those scores, it does give you your total, right? But that total is not accurate because it does not take the curve into account. So when I tell everybody, I'll post a message on the announcements on Moodle. If you don't get the emails, if you click announcements, there are messages under there that say stuff, all right? So you will see whatever your overall score is on Moodle, and then you can divide that, your total points, not the percentage, but your total points, add your two test scores together and divide it by, 180 or something. I'm just throwing that, that's not true, but by something like that, right? That'll take the curve into account. You can't, if I say, oh, there's, you get 7 points for this first test, you don't add the points and then divide it by lower score, or you've double curved yourself. That's a double bonus. That's maybe what you want, but don't do that because that score won't be accurate. All right. So you'll at least have kind of a ballpark. I can definitely give you some more information with that after Thursday, once I get those other scores, if you've got questions about it. But all of that stuff will post. I have to have scores posted by 5 o'clock Tuesday, not today, next Tuesday. And then it will all be available. So, but if you got questions before that, I can definitely help out. But so 210 should be the lab score, right? All right, any other concerns? And so I guess I didn't give you all of those details. The lab is 25% of your grade and the lecture is 75%. And so once you get your lab figured out and, say you've got a 90% in the lab, you take that 90 times 0.25. That's going to give you some decimal number. You take whatever you've done in the lecture, times that by 0.75. You add those two numbers together, times it by 100 if you want to see a whole number, not a decimal. Either way, it's a percentage, right? And that's where you will be. Okay. So with that said, hopefully you guys are attending lab. You're doing decent in lab. Lab definitely can help bump that score up, but it also can drag it down if you're not going. I think this semester I've had no feedback with people just flat out saying I'm not going to lab, right? But I don't know for sure, right? If you're in that boat and have missed a handful of labs, well, that's kind of bad news. You need those points, right? Okay. We're shifting gears, but sort of not, as we were talking about some of the details in the last unit of material. This next chunk kind of expands upon that a little bit, and we start thinking about some more details about DNA. We want to think about how, we've talked about the genes and how they have the potential to be inherited on 2 different chromosomes, and it depends on which one you're getting as far as the information that either of those might be contributing. And we talked dominant and recessive. We kind of mentioned good and bad copies with some of that stuff. Now we want to think more details about the information itself, the DNA, and how we go about in ourselves or potentially in a laboratory situation, how we can go about and replicate this information. And it opens up all sorts of potentials when we think about molecular biology and genetic engineering. There are lots of possibilities. Any of you guys concerned about or try to avoid things like genetically modified foods? Silence. So no, you don't care. I just want a sandwich. Okay. So sometimes there are potentially, some thoughts or concerns about some of these things where products have been manipulated, incorporating things into them that they've never had before. Oftentimes, from my standpoint, I think of benefits and promise from genetic engineering, right? I can tell you, I'm a microbiologist. I think about things like this, and oftentimes in that realm, we always think about promise. We think about how good these things are and the potentials we have to do all sorts of things, not just talking about food stuffs, all right? Just understanding the world around us through the ability to understand and manipulate genetic material, right? You can take a gene from something and put it into something else and have that something else express it, and you can get information about it. So you can manipulate some organism and maybe put it into something super easy to grow, like a bacterium, and you can get a tremendous amount of information pretty quickly, all right? And that's lots of the stuff that we think about. And really, it wasn't until I had started teaching this course that I started thinking about stuff like food, and thinking about how things like that get manipulated. And maybe it's not always good stuff, all right? There are some instances where maybe a product gets manipulated, some sort of food stuff, and maybe, you know, they don't have to tell you if it's been modified. Sometimes they do. They tell you if it hasn't, and they charge you more money for it, all right? Deep down, there's probably not a lot of difference between the two, but sometimes there could be, all right? There could be some product that's been manipulated, and now it's got a protein within it from some other thing, maybe a tree nut, and you happen to be allergic to tree nuts, and you're drinking some soy milk. And you think soy milk, that's coming from bean. That's all fine and good, but it's also got something else in it. And you drink that stuff, or you eat some tofu, and potentially have a severe allergic reaction because you didn't know, all right? It kind of makes me wonder, as you look at some of these things, sometimes you see labels that say, oh, this product was processed in a facility that also processes whatever, tree nuts, eggs, dairy, things like that. That gives you some allergy information, but it's also kind of just covering their bases. Maybe something's modified and there's something in there that is part of that product. They don't have to tell you that, all right? That's what gets me, is the lack of transparency. The fact that they're not telling you all of this information, I think that's not fair, all right? Not, you know, some of this stuff may cause harm, and you should know that, all right? So deep down, most of the things that we consume as we go to the supermarket, there's been some sort of manipulations, right? And when we think about just agriculture in general, we have been manipulating things since we started agriculture in the first place. As we start saving seeds from plants, this plant was more robust. It had a greater output. We save some of those seeds and we plant them the next season. And now those plants are better. And we keep progressing generation after generation. We've been doing this stuff, just not in the lab. It's been happening in the field, right? Where we have been able to control and manipulate based off of the whatever we feel is important, like a higher yield in wheat or something like that, right? But then we take it a step further and go into the lab and alter things differently, right? Whether that's good or bad is kind of up in the air. Again, most of it, I think, is probably harmless, right? So as we start, we want to show a little picture of Dolly. Dolly was the first cloned mammal, all right? Cloning, you know, there are all sorts of things that we think about with cloning, and it has been happening for quite some time. When we think microbiology, bacteria, we can manipulate, we can do all sorts of things. When a bacteria replicates, truly, they're like clones of one another, and we've done no sorts of genetic manipulation, right? We can create recombinant bacteria where we put other information into them. It's similar sorts of an idea, right? Stepping it up to mammals was a huge, huge feat, all right? It took a long time before this actually was able to happen, all right? There were numerous failures, right? But they kept trying, right? And part of that, you know, it's not exactly the same, but it kind of makes me think a little bit like this, all right? If you think about sexual reproduction, all right? Oftentimes in the process of intercourse, you do not produce an offspring, right? It depends on the circumstances. It depends on the types of animals, but even sheep, they may require multiple breeding sessions before they're actually impregnated, all right? It truly kind of depends, right? So, and deep down, that's probably pretty fortunate, all right? If every time intercourse occurred, you had a child, you've taken all the fun out of it, right? Not so good, right? So, couple hundred attempts before this actually happened, all right? And a lot of it was what's happening with this egg and trying to get everything to happen properly, all right? So we will come back and think a little more details about cloning as we progress through here. I'm not going to go off and talk about all sorts of stuff right now till we've kind of given some details about the molecular biology in the first place, all right? But as we've got her up here, basically, real quick, what we did to get Dolly, all right, was to have two sheep, all right, both females, all right. One sheep donates an egg, all right, and in that egg, we have a nucleus, and that nucleus has chromosomes within it, all right. Diploid or haploid? Single egg. It's now been fertilized. Diploid or haploid? What do we got in this egg? Haploid. One copy, right? Once it's fertilized, you get two copies, right? So you got a haploid egg, right? It's only got half the instructions. It needs to be fertilized to become diploid, and then we would start developing a sheep, all right? So we take an egg from a sheep, right? And as it turned out, the sheep that we took the egg from had a little black face, right? So we take the egg from that sheep, and then we take a cell, diploid, from another sheep, all right? That diploid cell, we take the nucleus, and we are going to put it into that egg. We took the nucleus out of it. I skipped ahead. We took the nucleus out of the egg. So now it's just a shell, if you will, all right? And once we took that out, we've had the nucleus from the other sheep, the sheep that we're going to clone, that looked just like this, all right? We took a mammary cell, had the nucleus, put that nucleus into the egg, gave it a little shock that stimulated cell division. And once that happened, that now behaves not like a mammary cell, but like a fertilized egg. And it starts replicating and generated Dolly, all right? Dolly was born. She died pretty young. How did she die? She died of old age, right? That makes no sense, right? But it does, all right? The sheep that donated the mammary cell was already middle-aged, right? She was already old, and her DNA had already gone through multiple rounds of replication. It had already done lots and lots of work, all right? And we took that cell and we put it into this egg, and now this newly forming embryo is already old, all right? That's kind of crazy, all right? And if we think about that type of an idea and, trying to clone humans, right? It's probably not a very good idea for me to take some of my DNA and try to clone myself because I'm already old, right? Super old if you're talking to my kids, right? I'm already old, right? I was born in the 1900s, right? That's what they tell me. I'm just like, get yours, right? So there's issues, right? And it kind of begs the question, sort of makes me think, if you wanted to clone a human, the best thing you could do is take some DNA from a baby and then clone it, right? If you just hatched out a kid, I bet you the last thing on your mind is, oh, we ought to clone this and get another one right now, right? Probably not what you're thinking about, all right? But that would keep stuff all basically young, right? There are definitely some potentials for maybe we can manipulate some of these things and change that, all right? So she had all sorts of diseases. that you would see happening in an older, well past her prime sheep, but she was young, right? As far as when she was birthed, she was still young, but she wasn't, all right? And that's kind of interesting to think about, all right? And with that said, we're going to talk about our DNA and the telomeres, the ends of our chromosomes. And that becomes super important when we think about some of these details, all right? Indicating kind of the age of that DNA, right? And impacting things like this, okay? So we'll see a little more about that. So initially, we need to think a little bit about DNA in the first place, right? There were all sorts of individuals that were trying to figure out what is the molecule that contains the information that governs us, right? That governs life, right? The two thoughts were proteins and DNA, right? Which one is it, right? There is a boatload of variation when we think about proteins. That seems like that could be pretty good, right? Maybe that's the information, right? There's all sorts of different things when we look at all of the varied types of proteins. Maybe this is the information, right? We know DNA truly is, all right? But that was the idea, right? DNA has a specific amount of bases and they appear randomly assorted in there. They are not, right? Variation, right? So a handful of different scientists that were really kind of racing, kind of battling against one another to figure out what is this substance? What is it that is the material of life, all right? And there's, there's a lot of stories about how this got discovered, a lot of detail that I think is pretty fascinating, right? We see a handful of them here, the ones we truly give a lot of credit to, but there were others, right? There were some very well-known chemists that were trying to figure these things out. Linus Pauling, a multiple Nobel laureate, was on the cusp of figuring these things out, all right? But not, all right? We give credit to Watson and Crick, all right? They did figure it out, but they also were kind of thieves, all right? There were a few issues with this, all right? So, and we can see McCarty here, geneticist, Watson and Crick. Rosalind Franklin is not pictured in this image. We'll give you a little bit of background with that, all right? So, and I'm not 100% if it was McCarty right now, even though I'm seeing him up there. Rosalind Franklin, and maybe it was McCarty, maybe it was, I could be wrong. She's working with this guy, right? Watson and Crick are working together. Oftentimes, when we think about science, sometimes it's good to talk with other people. Oftentimes, we may find ourselves at the pub having a beverage and talking a little bit of science, right? Talking shop, right? Talking about things. And as it turned out, Watson and Crick, they're having a beer with McCarty, maybe, right? And they're talking about some of this stuff. And he lets on that Franklin has been doing some X-ray crystallography. And you can take proteins, you can take molecules and shoot X-rays at them. And the diffraction pattern, as those X-rays hit the molecules, causes the rays to scatter. As it develops on the film, it gives you a pattern, all right? A diffraction pattern. right? I don't know as I look at these, what it means, because that's not what I do, all right? But as you see some of these diffractions, it gives you some ideas about structures of molecules, right? We talked about molecules and like Tinker Toys and building them together and making some structure, right? So you can visualize it three-dimensionally, right? This gives you kind of a similar sort of an idea, but it's a two-dimensional plane. It's, you know, a sheet of X-ray film, right? So anyways, it lets on that she's got some diffraction patterns from some crystallized DNA molecules, and they buy him another beer, right? That's my idea. And then maybe one more. And they finally persuade him, let's go back to your lab, right? And as they get to her lab, I envision that old Crick opens up the drawer, finds the crystallography, takes a quick look at it, puts it back in the drawer, and they race back to the lab. He had been doing some of this stuff. He hadn't been doing it with DNA, but he knew a bit about diffraction patterns. He had some ideas, all right? And as he looks at this, it was like this epiphany, this aha moment. He sees it and is like, holy cow, it's a double helix. Because they have no clue what DNA looks like, right? That's what they're trying to figure out. They're trying to figure out all of this stuff. What is the molecule that contains all of this information, DNA or protein, and how is it? How does it look? We've got no clue, right? He sees this picture, done deal. It's a double helix. They race back to the lab and they start with their modeling, you know, things, the Tinker Toys. They start building this structure, the double helix, right? And in turn, Nobel Prize, right? We give them credit for solving it, right? Rosalind Franklin got ripped off, right? So technically, you know, and fortunately, we mention her, we talk about her. She did not receive the Nobel Prize because she died before it was awarded. And apparently when that stuff, you can't give it to a dead person, right? Is the rules. So she should have got the Nobel Prize. She gets credit, but there's lots of times, there's lots of biology books that she's kind of just glossed right over, all right? They don't talk about her. And I think that's truly unfortunate because she did a lot of the groundwork that truly sparked the idea in Watson and Crick as they kind of, you know, looked in there when they shouldn't have been, right? And saw those details. So they knew some information about DNA, but like I said, they didn't know how it was all assembled and that it was a double helix, that it was connected like that, all right? So all sorts of chemistry, all sorts of ideas with that as far as, you know, building stuff and thinking how it fits. They knew phosphates and sugars and bases, and there's a handful of bases, but how is it that they interact? And that ultimately, with the diffraction patterns, was what opened up those, you know, opportunities to understand it. So every DNA molecule is made up of the phosphate, sugar, and base, and it's the base that varies, all right? There are pyrimidines, a single ring structure. You don't need to get bogged down with all of the letters and whatever, but pyrimidines have a single ring and purines have a double ring, all right? And as we have them listed here, they pair up this way: a pyrimidine with a purine, a C with a G, and a T with an A. That's always how they pair. And we call it complementary base pairing, all right? C and G are partners, all right? And A and T are partners. And this also provides kind of a insurance policy at the cellular level to make sure that we don't mix up and try to pair, you know, an A with a C or an A with a G, for that matter, all right? It doesn't work, all right? They can't pair like they truly should, all right? So we will see this in 1 second, all right? You've got this with one of these bases, and on the opposite strand, you're going to have the same thing, but the complement, the other base, all right? So always a pyrimidine pairing with a purine, all right? And C's with G's and A's with T's. That's how it happens, all right? And this is the information, all right? This assortment of these nucleotides, all right? And all three of these things, the phosphate, the sugar, and the base, is what we call a nucleotide, all right? And as they incorporate, we start talking DNA, right? RNA molecules, we'll mention a little bit later, are very similar. The sugar is a little different. It's got another oxygen on there where this one's missing it. And there is one different base. Instead of a thymine, there's a uracil, all right? But otherwise, it's pretty similar, all right? And that will be some of the ideas with proteins and what does this information mean. I believe that's the next chapter. So there are, as we move through here, some details that are, well, pretty complex, right? And hopefully as we walk through here, it makes a little more sense, all right? As we look at the double helix, it's kind of like a spiral staircase as it's twisted around, all right? The sugars and phosphates are the light blue ribbons that make up the backbone of that staircase. And the colored bits in the middle, these are the nitrogenous bases, the adenines and thymines, guanines and cytosines, right? And with that, they make the steps, or if we think of it like a ladder, they're the rungs of the ladder, right? Stuff you can step on, right? And as they pair together, the structure twists, forming this double helix. You can also see a couple of numbers at the top and at the end, all right? And it's the orientation, as we look at it right here, blown up, of these sugar phosphate groupings, all right? We can see the sugar phosphate here is 3, and the one it pairs with up above, 5, it's this carbon that's available, 3, 5. And on the other side, it's backward, all right? 3 at the top and 5 at the bottom. We call this anti-parallel. It's going in opposite directions, all right? So that also is important when we start thinking about the information, because when we read it, we want to read it in a specific direction, all right? So as these guys all pair up, it's 3, 5, 3, 5, 3, 5 as we're moving up here, right? 3 to 5, and the same here, 3 to 5 as we're going down, right? Inside those rungs of the ladder, the complementary nucleotides or the complementary bases, the thymine with the adenine, they pair and they form hydrogen bonds. If you guys remember from clear back in chapter 2, I believe it was, we were talking about different types of bonds, and covalent bonds are super strong, right? Hydrogen bonds are the weakest types of bonds, all right? Everything else, the phosphates, the sugars, the nitrogenous base, they're all linked together by covalent bonds. This one nucleotide is very strong, right? As it's all linked together. But holding the rungs together, the thymine with the adenine, they hydrogen bond. So they don't form a super strong bond. It's a very weak bond. And that's very important for DNA. It doesn't matter if we're thinking about DNA that's getting replicated to make more DNA for the cells, you know, for mitosis to just create a new cell. Or if we're thinking about opening up that DNA to read the instructions and build some proteins. It's a lot of work to unzip that molecule, right? To separate it apart. And if it was all covalently bonded together, it would be too taxing, too much energy would be needed to rip those molecules apart. But a hydrogen bond can be broken very easily. It's like a zipper that can easily zip up and unzip, all right? So A's and T's form 2 hydrogen bonds, and the guanines and cytosines, the G's and C's, they form 3 hydrogen bonds. That's another mechanism to ensure that they have matched up properly. And if you get the wrong base in the wrong position, you don't hydrogen bond like you should, all right? So there are proofreading mechanisms when we're copying the DNA that ensures that all of these guys have bonded like they should, right? And if somebody winds up in the wrong place, the mechanisms recognize that and they correct it before it's carried on, right? Almost always, right? There are potentials for some things to not get corrected, but usually it does, all right? Especially things like this, all right? These typically, we know if we don't pair properly, all right? If there is a change, and now in this one strand, there has been an alteration, and maybe now this is a cytosine on this side instead of a guanine, and as we're replicating, we put a guanine over there, it has altered, but it still pairs properly, right? So the proofreading mechanisms don't recognize that as an issue, but maybe the protein that it should be making does, right? That could be a problem. And mutations happen when we get shifts and changes in that coding information. So there are a handful of things, and I kind of hinted a little bit as we showed those nucleotides, differences between an RNA or a DNA molecule. So this is just the sugar molecule, ribose and deoxyribose. We've talked about also thinking molecules and macromolecules. We talked about sugars. We talked a lot about glucose as we were thinking metabolism, right? Ribose and deoxyribose, these are both types of sugars, but this isn't the stuff that you're sprinkling on your cereal, all right? These sugars are 5-carbon sugars, and they serve as backbones or scaffolding for other molecules to link up onto them, all right? They're not sweet tasting, all right? It's different, all right? So the deoxyribose on this number 2, so we've labeled the carbons here, 1 through 5, 5-carbon molecule. The deoxyribose on the number 2 carbon just has a hydrogen, right? And on the ribose, it has the oxygen, right? Deoxy is without oxygen, all right, is deoxy. So one difference, right, are the sugars. So there are, and we've already kind of mentioned a little bit about this as well, there's a lot of differences. When we think about prokaryotes and eukaryotes, right? There's also some similarities. As I mentioned, you know, they're still doing the same sort of stuff, pulling in nutrients, metabolizing those nutrients, replicating their DNA, and dividing, right? These things happen. In a eukaryote, we've got a lot more business going on the inside, where we have taken the responsibilities and walled them off into different compartments or organelles. In a prokaryote, we don't do that, right? So in a eukaryotic cell, and it's also considerably bigger than the prokaryote, we've got a membrane-bound nucleus, and inside of that nucleus are lots of chromosomes, and they are linear, right? Big, long strands of DNA, like a spaghetti noodle, kind of, right? And over here, I don't know why it looks like it is linear, but the prokaryotic chromosome is circular, like a rubber band. There is no membrane-bound organelles. There's no stuff surrounding it. It is free to be accessed at any time, right? And when we talk about making some proteins, that becomes super important, right? It's very different with a bacterium compared to a eukaryote. There's a lot more stuff going on, all right? And we also need to think a little bit about that when we think about genetic engineering, all right? If I want to take a gene from me and put it into a bacterium, I can't just take that piece of DNA and slap it into the bacterium and expect that the bacterium is going to be able to use it. I have to manipulate it first to take away some of the information that we have in our DNA that bacteria don't have, all right? We got some extra stuff, all right? Some spacers, some stuff that maybe has been called junk DNA, all right? But it's really probably not junk, all right? The prokaryotes don't have it, all right? They're bare bones and they don't need all of that extra detail, all right? So in a prokaryote, the chromosome is kind of in an area that we call the nucleoid, but it does not mean it's a nucleus. It's just sort of a region kind of central to the cell where that material is. All of the little red dots or orange dots in here, these speckles inside of the prokaryote, these are all ribosomes. And this is where protein synthesis occurs, is on a ribosome. Over here on the rough endoplasmic reticula surrounding the nucleus, we also have a bunch of ribosomes. There are some free-floating ribosomes in here as well. Dependent upon the type of protein that's being generated, it will depend on where it's synthesized, all right? We'll hit some of that in a bit, all right? So in us, or in eukaryotes, the DNA, again, it's linear, all right? A lot of information, all right? Our cells have, in every nucleus in our cells, there's about a meter's worth of DNA, right? Which is mind-boggling to think that, you know, we've got trillions of cells that make up our body, and each of those having upwards of a meter long worth of DNA in it. How does it fit into those cells? How do we get it all in there? And compaction is the answer, all right? Some of this stuff gets more compacted as we think about the synthesis phase, when we're trying to replicate the DNA and then moving into mitosis or meiosis, it's compacted even tighter, right? And then when we're back just into interphase, it's still got some compaction, but it's a little more open for business, if you will, right? Able to be read. So we think of it as this double helix, this linear molecule, but in a eukaryote, it's got other things that help to kind of keep it together, all right? So there's proteins that are called histones. They're like little spools, and the DNA is like thread twisted around those spools, all right? It helps the DNA to be contained. It helps with the compaction. It ensures that in the process of replicating, the DNA doesn't end up getting broken into fragments, all right? If you think of it like I described, like a spaghetti noodle, all right? If you've got a spaghetti noodle that is a meter long, all right, and you are trying to replicate that and separate it, and it's just two big, long, meter-long noodles, and you're trying to pull them apart, the potential for those noodles to break is pretty strong, right? Highly likely that we're going to have some damage, right? But if we twist them around a bunch of proteins and we compact it and sandwich it all nice and tight together, and then we try to pull it apart, now the possibility of that stuff getting broken apart is almost non-existent, all right? Because it's just all wound up super, super tight, all right? So the DNA right here twists around a histone, and there's a handful of proteins that make this little spool, all right? That starts wrapping around it, and it continues to wrap around all of these proteins, all of these histone proteins, and each little bead, like a necklace, each little bead is DNA twisted around the histones, and we call those guys nucleosomes, all right? The little beaded DNA protein complex, right? And as they're all twisted around and kind of held together, we call this chromatin, all right? DNA twisted around its histones, all right? As we are in this part here, this is where we are able to be thinking about like interphase, just doing our business, being able to be accessed and read. And then we shift it over and start thinking about the separation, mitosis or meiosis, right? So it condenses further as we twist and pack it up tighter into this chromatin, and then as we've replicated it, now we've got two copies like this linked together with the centromere, right? Held together, and that is what we see at the end of the synthesis phase, moving in to mitosis or meiosis, either way, depending upon the types of cells that we're talking about, right? And we pull them apart, right? And hopefully you remember in mitosis, they all just line up, all 46, and we separate the sister chromatids, right? And in meiosis, the homologous pairs, two chromosome number ones, the sister chromatids and both numbers one pair up together, creating a homologous pair. And we separate the homologs, and then we separate the sister chromatids, 2 rounds of division for meiosis and 1 round for mitosis, right? Once that has happened, then we go back to this, loosened up a bit, ready to be accessible and read, right? If that's what needs to happen, right? So this, as I described, there's complementary pairing A's and T's and G's and C's. You can also see the sugar phosphate backbones, the anti-parallel nature. The arrows are trying to tell you we're going in opposite directions, right? Because as we think about it, DNA and this bit, it's this side versus this side. 5' to 3'. The arrows pointing to the direction that we would be laying down these molecules. 5 to 3, and it's the same this way. 5 to 3, right? Sugar phosphates as they're linking together. That's what we're talking about, right? The carbons that are linking them up together, right? So we've also drawn these with different shapes in here to signify how they pair, so you can see this. And I think you'll do some stuff like this in the lab, probably coming up, or did you already do some of this? You already did it? Oh man, I'm behind. I'll start talking real fast, get you up to speed. Okay, so you've already kind of looked at this, and hopefully you know a bit about the complementarity of these nucleotides. So we know, because of research, we know how this DNA replicates, right? There were a handful of ideas as far as what happens to these molecules. And through testing, by labeling with some radioactive nucleotides, we're able to follow this process as it moves through to see what happens, who goes where, right? We know it's a semi-conservative process. So this is the parental strand. This is your 1 copy of DNA. We're going to replicate it. This is the synthesis phase, right? We're getting ready to make new cells. We're going to go through mitosis. And here, we've done it. We've got 2 new cells, right? We're not showing you all of that stuff. We're just showing you a couple of rectangles, right? This is the parental cell, and these are the 2 new daughter cells, right? The blue stuff is the new. The gray stuff is the parental, right? And semi-conservative means the DNA unzips, and both sides of that DNA, that parental DNA, both sides serve as templates for the synthesis of the daughter strand, the blue guys, right? Or the blue girls, call them daughters, right? So these will complementary pair, and basically this blue one right here is the same as this gray one right here. And this blue one is the same as that one, as they have complementary pair, right? So unzipping, serving as templates for the daughter sequence to lay down, complementary pairing, and now each of these even though we've got different colors on them, each of these sequences are exactly the same as the top one, right? It's the same stuff, right? That's what you want, right? It makes sure that we keep things consistent, right? So there were other ideas, that the DNA was conservative. It would open up, and once it opened up, you'd get one of them that was both blue and the other one was both gray, right? You never lose this. It's always stays together. That's not what happens. We see this, right? The other was called dispersive, where it's fragmented, and you have both of these that were, some of them have some blue on both sides, some of them have gray on both sides. It's all just fragmented, chopped up, and replicated like that, kind of a mishmash. It doesn't do that either, right? Through the radio labeling with the nucleotides, they were able to witness and see it over the course of a handful of replications, how it was occurring, right? And that is how we know it to be this semi-conservative, right? Two template strands, right, if you will. This lends to some confusion, right? Definitely. There are ways that it replicates, and it's not the same on both strands. Although looking at it, part of it, you see parts are the same, right? So on this, what we're looking at is the parental blue stuff and the new stuff getting laid down in the middle, all right? So this origin, this black line right here, there is a specific piece on that DNA that's basically like the starting line, right? This is where it will all happen, right? And it's an area that different molecules that are going to help with with DNA synthesis, they recognize and link up there, and it's where it all starts this process, right? So we call it the origin or the origin of replication. It's, again, the starting line, right? So with that said, the DNA molecules are in opposite directions, right? And the polymerase, technically, it only really wants to go in the forward direction, right? Going in reverse doesn't work so well, all right? So there are some differences with this, all right? And we can see the strand. So 3 to 5, 3 to 5, as we're looking at this, right? And as we are reading, I'm going to start with this top one. This first bit, that's the solid arrow, right? This is the forward direction, right? And so we are reading the DNA, the parental DNA, that is going in the 3 to 5 direction, and we start copying 5 to 3, right? We're going toward the 3, right? And the same down on this strand, right? Both of these happen, and that's the bit that's doing it properly. The other two that are going away, so these replication ports were unzipping the DNA. This is as it's being unzipped, and this is like the little zipper, it's what's doing the unzipping. Helicase unzips, breaks the hydrogen bonds. This moves toward the zipper, and this is moving away. All of these little discontinuous fragments we call Okazaki fragments. The other strand, the lagging strand, the one that's going slower, it lags. It has to be replicated in little stutters, little teeny bits, right? Discontinuous pieces. And each time, the green guys up here are a chunk of RNA molecule that allows the DNA polymerase, which is what's going to be laying down these red guys. It allows it, it's like the on-ramp to get onto your interstate, right? It allows those molecules, the polymerases, to link up. And if you're going in the right direction, you only need one on-ramp, right? But if you're going in reverse, we can't do it quite like that. And so it's little bits of fragments that are laid down. And in between each of these is a gap where they're not fused together, sugar phosphate, right? We go back and fix all of that, right? So it's not just a smooth process. And we see it happening dependent upon where the origin is. On opposite strands, there are two bits that we would call the leading and two that are the lagging. And it's dictated by the starting line, right? And once we start, and it's a linear, this is a linear piece of DNA, our starting line is right in the middle, and we've got people running this way, we've got people running that way, right, on the specific leading strands. And then we got them kind of moving a little slower as we're trying to catch up oppositely, right? I know this is a little bit confusing, right, as we move through. This yellow band is the polymerase, the DNA polymerase that's allowing these nucleotides, the new ones, to bond, right? And it's a lot slower on the lagging strands. That's why we call it lagging, right? DNA ligase, which isn't up here, but it would be participating in between each of these Okazaki fragments. There's some other stuff that happens too, right? Another polymerase comes in after it's been started, and we're doing some more road construction. We chew all of this green off and replace it with red, but it's not the same as this guy. It's a different polymerase that comes in a little bit after. And it's like, oh, green stuff, I want to get rid of that. And it's going to do the same over here repeatedly, right? In between each of these, the ligase, which is like molecular superglue, it comes in and forms a covalent bond with the sugar phosphates that didn't get linked together in between these separate fragments. Now they're a continuous piece, right? So much stuff, right? Okay. The ends of these chromosomes were like here, the little tails, if you will, right? If this was an entire intact sequence of DNA, right? The ends of the chromosomes are maintained by a telomerase enzyme, right? And it kind of depends on these circumstances here, right? The telomeric DNA are like big, long repeats of nucleotides. It's like the information, it's no genetic information that says, oh, make this. It's just like a protective coat, if you will, that helps to ensure that we're not damaging the DNA, all right? So there are issues as we move through here where this lagging strand, as it's laid down, there's a little bit where the primase isn't able to come in, and primase is what's going to put the RNA bit down on there. It's not able to put a little piece on there, all right? So now you've got a chunk of telomeric DNA on one strand, and there's nothing for it to complementary pair with. That's going to break off, right? This, deep down, as we're looking at this process right here, this is aging, right? As our cells replicate, most of our cells have the ability to replicate 70 to maybe 90 times, depends. Some cells maybe a little bit more, and a few cells don't have as much, and they, like maybe nerve cells, right? We don't see them replicating in the first place. They kind of just stop, right? But once we move through all of that telomeric DNA and get to the business of the chromosome, that cell's going to die, right? And it ultimately is a pretty good picture of the aging process. Can you slow this? Can you make it so that it doesn't happen so rapidly? Could we potentially have found a fountain of youth? The telomerase is an enzyme that puts on this information, and we don't think about normal cells having telomerase activity like some other cells do. Cancerous cells are rich with telomerase activity, the enzyme that's going to bring this stuff down. Cancerous cells are immortal. They're cells that don't know how to die because they never run out of telomeric DNA. Every time they replicate, it's like they are a brand new baby cell, right? Over and over and over, right? Countless times because of this activity, right? Normal cells don't have telomerase activity, but you have telomeres, right? Not the A's is the enzyme, right? And so that sets the stage for issues, right? As it's unable to be replicated, right? And like I said, 70 to maybe upwards of 90 times that it can replicate, right? Before we start hitting the actual information, right? So Elizabeth Blackburn in 2009 received Nobel Prize. She discovered the activity of telomerase and how it can go in and lay down this new information in cancerous situations. And again, thinking, could this be a possible fountain of youth? Would you want these cells that have replicated to get this information laid back down? Maybe, right? Maybe that could be good. If they are cells that have been messed up, they're, some sort of crazy mutations have happened, and now they're not making the stuff that they should. Well, you definitely don't want those to keep going, right? So now we've got two different ideas. Do we want that for normal cells? And how in the hell do you stop it in cancerous cells, right? Two different thoughts for two completely different ideas. One, I want to live forever. And two, I don't want cancer to kill me, right? How do you work it, right? It's interesting, right? It's definitely interesting, all right? I think with that, I'm going to quit just the littlest bit early. It seems like a pretty good stopping point. We will talk about some of the proofreading and some of those details Thursday. Any questions or concerns? All right, guys, go dust the snow off your cars. If you're at home, you better start shoveling snow, dang it. Okay. Have a good day, guys.
Have a good day.
Thank you.